Dear student, welcome to Knowledge Quest. The subject is biology and I'm your teacher Lydia Asiago. Today we want to learn about plants, and in our last episode we discussed about plants and we have observable features that we are able to see in plants. We also discussed that most plants are differentiated into flowers, stems, leaves, and even roots. So today we want to measure on the structure of a flower being one of the organs in plants. So welcome to the lesson. We are discussing biology, and our topic today we are discussing on plants. Okay. Now, when we talk about plants are differentiated into flowers. Just as I said, plants, they are differentiated into flowers. Okay. Plants are differentiated into leaves. Plants, they have the stems. Plants have the roots. Okay. Those are the parts of plants. So we say they are organs in plants. Now, for today, I want us to measure on flowers. Now, how many flowers have you come across? The flowers that you've come across, are they of the same color? Are they appealing? Are they attractive? So with me here, I have a sample of flowers. And these flowers, they look very nice, they look very beautiful, they are attractive. They have different colors. Like now you can see this is a flower of a given plant. Of course, different plants, they have flowers of different colors. And we are going to understand why do they have different colors. So we have this flower that is yellow in color. We have some flowers, they are white in color. Okay. We have some flowers, like this plant has flowers that are, is that purple? Student, which color is that? Okay, I know you know the color. Okay, we have another flower that has this color. Which color is that? That is red. Okay, we have also another flower that has this color. So you realize that flowers, they are of different colors. And we are going to understand why are flowers of different colors. So just as I've said, today we want to measure on the structure and function of flowers. We want to measure on the structure and function of flowers. So structure and function. So, structure and function of a flower. Okay, so I want you to walk with me as we explore more on the structure of a flower. Now, you realize that a flower has different parts. So, I want to list the parts of the flower. So, parts of a flower. Number one, a flower has what we call sepals. Number two, a flower has what we call the petals. Number three, a flower has what we call the stamen. Okay, we are going to explore and learn more on what is that we refer to the stamen. And then we have the pistil. Okay. So those are parts of the flower, and we are going to see each part, and probably we give the function of each part. Now, I want to start with the first part of the flower that we call the sepals. So we want to start with the sepals. Now, plants have sepals, and you realize that flowers that have sepals at the young age, At the budding stage, those sepals usually they appear to be green. They look like leaves. Remember, leaves are always green. Most of them are green, not always, but we say most of the leaves are green. Green because of the presence of the chlorophyll. Now, when we talk about sepals at the early stage of development of a flower, that we say at the bud stage, the sepals, they enclose the flower. So I want to pick a sample from here so that we are able to see how at the budding stage, okay, let me pick this one. So you see like this flower, of course this is a leaf, but I want you to see that at this point of development of this flower is at budding stage, at this part. And you realize that the sepals, they are usually green. And when you talk about green, it means they have chlorophyll. Now, when you have sepals, these sepals collectively, we refer to them as the calyx. So collectively, we refer to the sepals as the calyx. So when you talk about calyx, calyx is a collective name that refers to the sepals. So we are saying that, number one, the sepals, they are green. They are green in color. Okay? And I've said when they are green, it means they have some chlorophyll. Then we are saying the sepals, they enclose the flower during the budding stage. So they protect the flower at budding stage. Okay? So they serve a role of protecting the flower during the budding stage. And we have said that they are green in color. Now, and I've said, of course, that collectively we refer to the sepals as the calyx. Now, you realize that there are flowers that have other special part, like now this one. This is a flower of hibiscus. This is a hibiscus flower. So if you check on this flower, you can see there is some green part of the flower. Now, this part that is green, this is what we refer to as the what? The sepals. But you realize that there are some special flowers that have other structures that we refer to as epicalyx. You can see, like this is hibiscus flower. It has this part that we refer to as the epicalyx. This epicalyx is also green, just like the sepals are green. So at times, it is good we understand that not all flowers that have the epicalyx, but we expect all flowers to have the calyx. Calyx, which refers to the sepals. And we've said that the sepals are green in color. I hope, my student, you can see that. These are the sepals. And if I was to count the number of sepals that I have in this flower, for example, I can say this is one sepal, this is 2, and then I have 3, okay? So you can count them. Remember, you can count the floral parts of a flower and use the floral parts of a flower to place plants into different classes, whereby we have the monocots and the dicots. So for this case, when I talk about the calyx, calyx refers to the sepals. Then we said that some special flowers, they have what we call the epicalyx, and both of them are green in color, and majorly their major function is to enclose the flower at budding stage. So in a way, they protect it from any mechanical damage or any invasion of disease-causing microorganisms. So I believe at this point, you can be able to see that most flowers, they have the sepals. So if I talk about this one, this is also another flower, okay? Can you see? These are a flower that is white in color, but it also has somewhat. You can see the green part, that is the sepals. Sepals. So these are the ones that protect the flower during development at budding stage. And you said that the sepals are green in color. I hope up to that point you are able to identify the sepals, which are also called the calyx, a collective name for the sepals. So you realize that most flowers, they have the sepals. And we've said sepals are green in color. Now we want to move to the next part of the flower. And I want to talk about the petals. So we talk about the petals. So we have the petals. Now, when we talk about petals, mostly we concentrate on these plants that are pollinated by insects, insect-pollinated flowers. Most insect-pollinated flowers, the petals are colored. So usually we say the petals are usually colored. They are colored. Why do you think these petals are colored? Now, let's first identify the petals before we confirm that they are colored. Now, if you check on this part, these ones. So if I can pluck this one, that is a petal, okay? So this is a petal. So you can see this flower has so many petals. And the petals for this flower, they are white in color, okay? Remember, I've said the petals are what? They are colored. They have different colors. And we are going to give a reason why they have different colors. So for this one, you can see these are the petals, okay? These are the petals. So anytime someone will ask you to identify this part of the flower, we say this is the petal. And these petals, for this flower, the petals are yellow in color. Now, we also have another flower. Okay, can you see this flower? Yes. This flower, the petals are these ones that are what? Are they red in color or they are pink? Maybe I'm not colorblind, or you are not. You should be able to identify the color of these petals. So you see the petals are what? They are colored. They are colored. Of course, we say for insect-pollinated flowers, the petals are colored to attract insects. You know, when we talk about pollination, the transfer of pollen grains from the anthers to the stigma of a flower is what we call pollination. And for us to transfer the pollen grains, then we have agents of pollination. And one of the agents is the insects. So insects, they are usually attracted to different colors. That's where you realize that flowers have, the petals have different colors. So you also have another color here. Can we say this is purple? Okay. So these are color, a different color in this flower. Okay. These are different color in this flower. So we say these are the sepals. Sepals are always colored. So if you want to identify the sepals in a flower, remember, sepals are always colored, these ones, okay? So like now you can see how many colors there. I have already 4 colors. We can add another color, okay? This is also another flower that has, which color is that, student? So you can see petals, they have different colors. Why are they colored? That is the question we should ask ourselves. Why are the petals colored? They are colored to attract insects, especially, or to attract agents of pollination, okay? So mostly for flowers that are pollinated by insects, you realize that their petals are colored. And another feature that we see in petals, these petals, number one, we've said they are colored. Number two, the petals are large and conspicuous. So we say they are large and conspicuous. Okay? So we say these flowers are, the petals are large and conspicuous. Like they can be easily seen from a distance. They can be easily seen. And of course, we say they are large. They provide a landing service for insects. They provide a landing service for insects. So we expect the insects to do what? To land on the petals. As the insects search for nectar, what happens? They end up picking the pollen grains from the flower. So in a way, we are saying all insect-pollinated flowers, they have petals that are colored to attract the insects so that the insects can carry out pollination. So that is what we talk about the petals. And then we've said they are large, they are conspicuous, they can be seen from a distance. Those flowers can be seen from a distance. Now that is about the petals. So we have said we have the sepals, the petals, the stamen, and the pistil. Now, having done the petals and the sepals, let's look at the other part of the flower. Let's check on the other part of the flower. Okay, so next we have the stamen. Okay, we have the stamen. Now, when we talk about the stamen refers to the male reproductive part in a flower. A stamen refers to the male reproductive part in what? In a flower. Now, which parts are these that represent the stamen? So at the stamen, this is where we have the anthers. Okay. At the stamen, this is where we have the what? The filament. Okay. So we want to see those parts of the flower that has the what? Has the male part. So the male part represents the anthers and the filament. Now, usually we say the filament, they hold the anthers in position. So these ones, they hold the anthers in position. Remember, the anthers are the one that produces the what? The male gametes, which we call the pollen grains. So if we were to check on the structure of the filament, I believe you can be able to see. So if you are very keen, you can see there is this part that looks yellow. Okay, can you see this part? Those are the anthers. Can you see the part that looks yellow? That is the anthers, and that is where we produce the pollen grains. Then there is this part that is supporting the what? This part that is supporting the anthers in position. This is what we refer to as the filament. So you can see, this is the filament. This is the filament. It is the one that is holding the anthers in position. So in a way, we are saying the work or the function of the filament is to hold the anthers in position. Now, when we talk about the anthers, what do? The anthers produces the pollen grains. Anthers produces the pollen grains. And usually you find that, especially for this flower, now that the petals are colored, we can say this flower is pollinated by insects. So you find that those flowers that are pollinated by insects, what happens? Their pollen grains, they're usually sticky. So if you take and have a hold of it, you realize that they produce a sticky substance. Why are they sticky? So that they're able to stick or to attach themselves to the body of an what? Of an insect. So we are saying for the anthers, they produce the pollen grains. Okay? Anthers, they produce the pollen grains. And pollen grains, I've said, they are the male gametes. Male gametes. They are the male gametes. So they carry the genetic material for the male part of the flower. So basically, that is what we talk about the stamen. So we are saying the stamen consists of the anthers that produce the pollen grains, and the filament, they hold the anthers in position. But now we need to ask ourselves, this is for, which plant is this? We said these are flower from hibiscus. This is hibiscus flower. So let me see another part of the flower, and we try to see where are the filaments. For example, let me try to see this one. So we said these are the petals, so we try to remove the petals so that we are able to see we want to see the filaments. Okay, so you see for this plant, for this flower, we have the filament. Okay, so for this one, we say this is the filament. Okay, this is the filament, and here we have we said we call it the anthers. The anthers, they produce the pollen grains. So, and you realize that for different plants, for different flowers, based on their agent of pollination, some the anthers, they are inside the flower, others the anthers are outside. So we are going to discuss that in the next episode, but for now, I want you to see that flowers have the male part, which consists of what? Which consists of the filament and the what? Filament and the anthers. So we can see that. Okay, that is for that flower. So that one we can see in various plants. You should be able to identify the male part of the flower and the female part of the flower. So maybe if I can check also over this flower we can also see it has so let me just try to remove the petals. We said those are the petals. So if I have the petals, you can also try to see the what? The filament. So the filament, these ones, at the end they have what we call the anthers. And it is at the anthers that we produce pollen grains that are sticky, especially for insect-pollinated flowers. So I believe up to that point, my student, you are able to see the male part of the flower. We are taking a short break. Stay tuned. Welcome back, student. Before we went for a break, we were discussing on the structure of a flower, and so far we have been able to identify the male part of the flower, which we said we have the anthers and the filament. So collectively, the male part of the flower, we refer to it as the stamen. So I want us to proceed to the other part of the flower that we call the pistil. Okay? So when we talk about the pistil consists of the female part of the flower. So we say this is the female part of the flower. This one we said is the male part of the flower. So the female is the pistil. So we refer to the female part of the flower as the what? As the pistil. Now the pistil consists of three parts. Okay, the pistil consists of the stigma. Okay, the pistil consists of the style. And lastly, the pistil consists of the ovary. Okay, the pistil is the female part of the flower and has three parts. And those are the parts that I want us to identify before we give the function of each part of the flower. Now, for this case, allow me to start with the stigma. Remember, I want us to first identify those parts of the flower. And now at this point, we want to identify the female part of the flower. And I've said the female part of the flower consists of the stigma, the style, and the ovary. So I want us to see, now if you check on this flower, you can see this part, okay? This is what we call the stigma. So this is the stigma, this is the stigma, this is the stigma, okay? So if I was to count the number of stigma for this flower, I will say this is 1, this is 2, this is 3, this is 4, and this counts makes it 5. So it means the stigma, they are in 5s. So we say when the floral parts are in 5s or multiples of 5, we say that flower belongs to the plants that belong to the class Dicotyledonei. So we are saying that this is the stigma. Now, for us to identify the style, then you will allow me to remove the other parts of the flower. Remember we said these are what? These are the epicalyx. So I'm removing the epicalyx. Then I further remove the petals. We said the petals are always colored. I want to remain with the female part of the flower. Okay. I want to remain with the female part of the flower. And I want us to identify the parts, the female part of the flower. Now remember, at this point, this flower have removed what? The petals. So I don't have the petals, but I have the sepals and the epicalyx. So we said these are epicalyx, and we have epicalyx, so allow me to remove them. Okay. Then we remain with the sepals. You'll also allow me to remove the sepals, so that we want to trace the part, the female part of these flowers. So these are the sepals that I'm removing. Remember we said the sepals are green in color, and they protect the flower at budding stage. So having the stigma up here, we expect to have the style and the ovary. So the style is at this point, and the ovary, we find it on the lower part. So I'm going to keenly dissect this flower so that we are able to see the female part of this flower. So I want us to see the female part of the flower. Remember, a flower is a living organism. Living organisms, they are made up of organs. So I'm trying to open so that we are able to get. Remember, we are looking for the what? We are looking for the style in a flower. Remember, we said the female part of the flower consists of the stigma, it consists of the style, and it consists of the ovary. So we'll try and get the style. We'll try and get the style. Remember, we are trying to check the style that is part of the female of a flower. It makes part of the female of a flower. Remember, the style, it is the one that exposes the stigma. So up to this point, remember, a flower is a very delicate organ, but you'll try and get the style. Okay. My student, I want you to see the style that has what? That exposes the stigma that has a sticky substance. So maybe I may not be able to remove the whole of it, but you can see from the inner part. So this is the style of a flower. I hope as you can get any flower and try to trace the style. So this is the style of the flower. I may not remove the whole of it, but I believe you can see we have the style of the flower. Okay. And remember, the style is the one that exposes the stigma. Remember, we need the stigma for pollination. So I hope at this point you can see what I'm trying to do. This is the style, and this is the one that is holding the stigma. Okay, it is the one that is holding the stigma. So up to that point, we can still try and get it. Remember, we are looking for the style, the one that exposes the stigma. Very good. So I think now you can see, okay? Remember, the style was enclosed. So this is the one that is holding the stigma in position. So you can see the stigma. And this part is what we refer to the style. Then remember, these are the what? We said these are the pollen grains. Pollen grains are produced by the anthers. So we expect the anthers to produce the pollen grains. And that's where you can see some of the pollen grains have stuck on the stigma. Remember, we said we are going to learn more on pollination so that we understand why will pollen grains stick on the stigma of a flower. So this is what we refer to the style. And you can see the style is very weak, but being enclosed in those tissues, it is able to expose the stigma so that pollination can take place. So I think for that you can check out with this. Okay, so it is the same flower, but I want you to check that the style is inside these tissues. It is the one that is exposing the what? The stigma. So I hope at this point you are able to identify the style. Now, before we give the function of the style and the function of the stigma, allow me to show you the ovary. Ovary, we said the pistil consists of the stigma. The pistil consists of the style. I believe at this point you have seen the style. So we want to identify the position of the ovary in a flower. Remember, the ovary is the one that produces the female gamete of that flower. So we'll proceed down here. We'll proceed down and we get the ovary. Okay. So the ovary, usually we find it at the base. It is in the ovary that we have the ovules. The ovules, they form the female gametes. The ovules, they form the female gamete. So we are trying to get the ovary. Okay. So I'll try to remove all these other parts as we remain with the female part of the flower. Okay? Now this is the ovary. Student, this is the ovary. So I've removed the male part of the flower and I'm now left with the female part of the flower. Now this is the ovary. Remember, the ovary was enclosed. It is not in the open, it was enclosed. Then this is now the what? This is the style. Can you see the style? Then at the end here, this is where we have the stigma. Remember, we are discussing on the structure of a flower. And at this point, we are discussing on the female parts of a flower. And we've said that the female part of the flower, we refer to it as the what? As the pistil. And this pistil consists of 3 parts. The pistil consists of the stigma, and I've said this is the stigma. The pistil consists of the style. This is the style. And you can see the style is connected to the ovary. So the female part, which is the pistil, also consists of the ovary. Now, inside the ovary, this is where we have the ovules. This is where we have the ovules. So if you were to open the ovary and try to observe the structure of the ovules, it is still possible. And of course, you can view it nicely when you have probably a hand lens. But let me try to open the ovary so that we are able to see what is inside the ovary. Okay? You want to see what is inside the ovary. And if you are very keen, my student, you can see there's something that looks like, don't know which term I can use, but we see the ovules. So, when we talk about the ovules, these are the ovules. Let me remove one so that you're able to see. Okay. So these are the ovules. So the ovules are the ones that form the female gametes. Okay. They are the ones that carry the genetic material of what? Of a female plant or a female flower. So in this case, we are saying that the ovary consists of the what? Of the ovules. So you can see them, and usually there are many. There are many. Okay. Usually we say the ovary is enclosed in an embryo sac. The ovary is enclosed in an embryo sac. So I believe at your own time, you can try and get a flower, try to explore the parts of the flower, try to see if you can be able to identify the female part of the flower and the male part of the flower. So at that point now, we can go back now and state the functions of those parts that we have identified. Now, we were able to identify the what? The stigma. Now, what is so unique with the stigma? We realize that the stigma produces a sticky substance. The stigma produces, so this one, it produces a sticky substance. Okay? Now, the question is, what is the role of that sticky substance? Remember, when pollen grains are transferred from the anthers, they should land on the stigma. So we expect as the pollen grains land on the stigma, we expect them to stick. As they stick, this sticky substance usually has nutrients. So the pollen grains usually absorb those nutrients and they germinate. So in a way, we are saying probably for reproduction to take place in plants, then the stigma has to produce a sticky substance that will enable the development of the pollen tube that will carry the pollen grains all the way to the ovary, because it is in the ovary that we have the female gametes, which are the ovules. So purposely, that is the function of the stigma. And of course, we've said the main function of the stigma is to receive. So if you are to give the main function of the stigma, we say, is to receive the pollen grains. And the features that enable it to receive the pollen grains is because of what? That sticky substance. So its main function, so if you are to be asked, what is the main function? The main function of the stigma is to receive pollen grains. That is the main function of the stigma, to receive the pollen grain. Then we move to the style. What do you think is the function of the style? Now that you are able to see the style, why do you think? What do you think is the function of the style? We say this is the style that is connecting the stigma and the what? And the ovary. So in a way, it is exposing the stigma for it to receive the what? For it to receive the pollen grains. So it exposes the stigma, hold it in position for it to receive the pollen grains. Then now we go to the ovary. When we talk about the ovary, this is where we have the pollen grain. We have the ovules. Sorry, we say we have the ovules. So it has the what? Produce the ovules. Okay. Now, when we talk about ovules, I've said ovules, these are the female gametes in flowers. These are the female gametes in flowers. So the parts of the plant, of the flower that produces the gametes, we say the anthers produce the male gametes, while the ovary produces the female gametes. And we are saying the male gametes are the pollen grains. The female gametes are the ovules. So basically, that is the structure of the flower and the parts of the flower with their functions. Maybe the last part that someone can ask, if you are able on what we have done here, are you able now to draw the female part of the flower? Are you able to draw the male part of the flower? That is the next question that we should answer ourselves. Can we be able to draw, now that we have seen, can you try to picture and draw it now in your book? How can I draw the female part of the flower? So if I was to draw the female part of the flower, then what will I draw? We've said the female part consists of the stigma, it consists of the style, and it consists of the ovary. So I'll just draw the stigma, probably. Then I draw the style. Then I will draw the ovary. Okay? And remember the ovary, inside the ovary, this is where we have what? This is where we have the ovules. So this is where we have the ovules. In the ovary, this is where we have the ovules. So I'm trying to draw. This is just a sketch. So if you were to draw that, you can draw. And remember, this ovary is enclosed in the receptacle. I think you need also to understand that the ovary is not just hanging. There is this part of the flower that attaches it to the plant. So let me show you. So like now we say, this is the flower, okay? If this is the flower is attached to the plant using a flower stalk, okay? We talk about a flower stalk. So we call it a flower stalk. Okay? So this part here, we have it as a flower stalk. So the next part we have that is now holding the ovary is what we call the receptacle. So this ovary is enclosed. So if I was to draw, then I should make sure that the ovary is enclosed and it is held by the receptacle. So let me try to do it. Okay. Then I say this is the ovary. Then this ovary now is enclosed in a receptacle. So we say this is a receptacle. Okay, so the ovary is not just hanging, it is held or it is held in position by the receptacle. So we say this is the flower stalk. So we say this is the flower stalk. Then now we have the ovules. So we have the ovules. Then of course now we say this is the stigma. Then we say this is the style. Okay. Then we say this is the ovary. Then we have the receptacle. Okay. So that is the female part of the flower. If I was to draw the female part of the flower, remember I'm doing a sketch, and being a student, I know you can do a good diagram. You can do a good diagram. Then now we have the male part. If I was to draw the male part, so we say the male part of a flower. Then remember we said the male part of a flower consists of the anthers and the filament. So the male part of the flower, so I can just draw. Then I can say so we say this is the filament. Then this is the anther. Then we say the anthers, this is where we have what? This is where we have the pollen grain. Okay, so if I point at one, we say that is the pollen grain. So basically, if I was to draw the structure of the male and the female, the best thing you do, you just cut, open the flower, and use a hand lens so that you can magnify the parts, and you do what? You draw the structure. And of course, anytime we say you draw a structure, you must do what? You must state the magnification. So you must state the magnification. You've drawn a structure. How do you state the magnification? So you must tell us the magnification for that diagram that you have drawn. You also must give us the magnification of the flower, the part of the flower that you have drawn. Now for the parts, when we talk about magnification, remember when you are given a specimen to draw, for example, I was drawing, let me say I was drawing this part, which consists of what? We said the stigma and the style. I may not draw the exact size, and that's why we are saying you must give us the what? The magnification. How do you get the magnification? Remember, you measure the length of the drawing, whatever that you have drawn. You also measure the length of the specimen that you have, and then you divide by the length of the drawing over the length of the actual specimen. So in this case, this is the drawing, and the actual specimen is what I have in my hand. So if I was to do magnification, I'll take the length of the drawing, then you divide by the length of the actual specimen. So you do the length of the actual specimen over the length of the, we have the length of the drawing over the length of the actual specimen. So once you do that, then remember the magnification has no units, you just give us the value. You subtract, you cancel the units, and then you give us the what? The magnification. Probably I can say times 2. Maybe this one can also say times 2. So magnification always has no units. So anytime you do any diagram in biology, remember to state the magnification. Now we've been discussing about the flower, and we have seen that the flower consists of various parts, and each part of the flower has its own function. So it is your duty now, go and identify a flower, probably at the garden, where you come across a flower, try to take that flower, try to observe the male part and the female part. I think you'll understand more, because it is an organ in plants that is very essential. It is a reproductive organ. Thank you for watching our show. I pray that in the next episode, we are going to learn more on plants and especially on the flowers. So always remember to stay tuned to our program, Knowledge Quest. The subject is biology, and I've been your teacher, Lydia Asiago.